academics as well as society. So far research is concerned, I am telling you, sir, my research is not very good. I am telling you. Of course, I did my...
Sir, now we are live, sir, on YouTube.
Okay.
Yes,
Okay, fine. So should we start, sir?
If you want to wait for 5 minutes so that at least 50 plus students are there, so that it will be nice.
Okay.
Within 2 minutes, sir. 11:15 is our time, so 11:12 or 11:13 is okay, sir.
Okay, then I can wait 2 to 3 minutes. Ujwal, sir, we can wait for 2 to 3 minutes. Let student to join and then we will start.
Sure, sir.
So, sir, when you finish your PhD?
Sir, I have completed my PhD in 2016. I joined Naresh Kumar sir's lab in 2011.
Okay.
Yeah, and before that, like I had a short stint of experience at Osmania University in genetics department.
Okay.
And later on, for a short period of time, I was there in Central Salt and Marine Chemical Research Institute.
CSCM.
And after that, I joined Naresh sir's lab in 2011.
Earlier, Dr. Jha was there also.
Yes, correct, sir. So I was, yes, correct, sir. So I was there at that time only. I was there for 6 months in 2011.
I did masters from LNMU Darbhanga, and he was there. He was there as a head of the department.
Okay.
Dr. Professor Jha. And thereafter he joined as a deputy director at CSCMRI. And I also came in Gujarat, 2003. In same year, sir. Sir had joined CSCMRI and I had joined NV Science College.
Yes, sir. It was always like, I have been able to, in my college days, I was able to listen to some of your lectures, and we have also interacted in past. It was always a pleasure to listen to you, sir.
Oh, thank you very much, sir. Thank you. Really, it's good.
Yes, sir.
Perhaps you came our college also with Naresh sir, if I am not, if my memory is okay, in 2013. No, that was another student. Okay, sir. एक बार सर अपने 2 स्टूडेंट के साथ आए थे, पर नहीं वो दूसरे थे, मुझे याद आया.
Correct, sir. So only thing that we realize in all these years that we have got so many experts and so many good researchers there in Gujarat. So it is always good to learn so many new things from everyone.
Perhaps Dr. C.G. Joshi is there. He is also doing very good work, sir.
Yes, sir.
Director GBRC. Earlier he was in Anand Agriculture University.
Correct.
And during our PhD, he was our sequencing project guide. Whatever sequencing work we did, that was only under his supervision. He also taught us some parts of genetic engineering. तो नरेश सर हैं या फिर जोशी सर हैं, ये लोग बहुत ज्यादा मतलब गुजरात के डेवलपमेंट के लिए दे कंट्रीब्यूटेड अ लॉट.
करेक्ट सर.
तो इनको अपन लोग बहुत रिगार्ड और रेस्पेक्ट रखते हैं इन लोगों के लिए, सर.
Absolutely, sir. Sir. So, sir, I just wanted to inform something.
Please.
So initially, I was supposed to take a lecture on deriving the structure of sugars, and what I thought that 1 hour may not be sufficient, because that also needs to be translated. We need to do certain practice. The students needs to get engaged. So what I thought is another interesting topic, that is, there are certain essential questions in nucleic acid chemistry. I was also listening to Naresh sir's lecture, and what I thought that if the stage is so high, so I need to jump with some more interesting topics. So I'll be taking the lecture on essential questions in nucleic acid chemistry, which is also a part of biomolecules only.
Very good. No problem, sir.
Absolutely, and that will be also pretty much important in terms of the exams. Certain trivial questions that probably we know, but we don't know the reasons. Like students don't know the reasons for most of them. So that I will try to cover in this lecture. Sure,
sir. Few things I had covered earlier. Yesterday, I also took lecture on that. but that will be little bit different. आप जो बोल रहे हैं वो थोड़ा अच्छा ही रहेगा, वो part missing जो है that will be entirely covered, so this will be better for a student. ये अच्छी बात है.
Correct sir.
I think we should start now.
Yes sir, Kundan sir, please.
So dear student, welcome back. Really, once again I feel pleasure to introduce one of the youngest teachers, academician and researcher, Dr. Ujwalkumar Trivedi sir. Dr. Trivedi sir is associated with Department of Microbiology.
Good morning, sir.
Yeah. कुछ कोई बोल रहा है? Yes, tell.
सर कुछ नहीं, उसने गुड मॉर्निंग बोला सर.
Okay, sorry. So Dr. Ujwal Trivedi sir is associated with Department of Microbiology and is coordinating MSc Biotechnology and PG DMLT courses at Marwadi University, Rajkot, Gujarat. He obtained his PhD in biochemistry from the Department of Biochemistry under Dr. Naresh sir. You already attended his lecture just before this. He is recipient of several national as well as international awards for his scientific accomplishment, including the Best Research Award at Gordon Research Conferences on Nucleic Acid held at USA 2015, awarded by Nobel laureates Professor Thomas Keck. Actually, he was discoverer of ribozyme catalytic RNA, I remember. He also received the prestigious Nature Publication Group Travel Grant Award for presenting his research in USA. Dr. Ujwalkumar Trivedi was elected as Fellow of Indian Chemical Society for his research on protein biochemistry and is regular speaker and panel member at various national and international conferences. He holds a keen research interest in chemical evolution, particularly RNA world, and in the emergence and evolution of protein. He is an affectionate teacher of biochemistry, molecular biology for graduate and postgraduate program of microbiology, biochemistry and chemistry. Dr. Trivedi loves developing effective mnemonic technique to simplify the teaching learning process. He is very curious as well as student centric teacher. So behalf of GSBTM and NV Patel Science College, sir, we welcome you on this particular crash workshop, and I am sure a student will take much information for competitive point of view and get benefited in all type of PG as well as PhD entrance examination. So I welcome you once again, sir, and now I am transferring session to you, sir. Please, over to you, Ujwal sir.
Thank you so much, sir, Kundan sir, Amit sir, and GSBTM for inviting me for this particular lecture. I hope students can benefit something out of this lecture. So my lecture would be like for two things. Definitely, it is going to be for competitive exam preparation, and another thing is that it will be also helpful to develop the thinking, scientific thinking. So I will just press.
कुंदन सर कुछ कनेक्शन कट गया क्या?
नहीं, कनेक्शन इज ओके सर.
तो सर का सुनाई नहीं दे रहा, आपको सुनाई दे रहा है?
हाँ, अभी सर प्रेजेंट कर रहे हैं, अभी आएगा सर, अभी आएगा. या सर, इज इट विजिबल टू यू, अमित सर?
यस सर.
हाँ, फाइन. Yes sir, Ujwal sir, it's visible. It's visible sir. Ujwal sir, it's visible.
Sir's mic is off.
Sir, kindly on.
सर उज्जवल सर,
एक मिनट में ऑन करके बोलता हूँ.
यस सर.
Ujwal sir, please on your mic. Ujwal sir.
Yes sir.
हाँ, okay.
I don't know, somehow it automatically got muted.
नहीं, it happens sir, it happens.
Okay sir. Okay, sure sir. So today, like, as I discussed earlier, so I'll be like touching upon some of the very, very focused basic questions, and which are also very essential in nucleic acid chemistry. So, from what I know is, Kundar sir had already given you basic information regarding the structure and functional aspect of nucleic acid, that is DNA and RNA. So we will jump a little bit more in certain things which are related to DNA and RNA, how a particular structure is like, causing a particular kind of function, why there are certain things present in the nucleic acids, all right, and why they are stable from many years. As I was listening to Naresh Kumar sir's lecture initially, he said that like there are certain processes which are constant from several billion years. So although like we are we are undergoing constant evolution, but there are certain processes which are very, very central, and they don't change. And in the same way, like this is true also for this very important biomolecule, that is nucleic acid, or in that matter, like DNA. So this is the outline of my today's lecture, that I'll be taking some essential questions, that why DNA replaced RNA in the evolution, then why thymine replaced DNA, why nucleic acids are never synthesized in 3' to 5' direction, why nucleic acids contains only pentose sugar, that is ribose, and not other sugars, and why DNA double helix is anti-parallel in nature. So this is like I regularly teach to my students when I'm teaching biomolecules to them. So this actually helps them to ask questions in science. They would not accept the information as such, but I'll just try to inculcate the art of asking questions. So I've just put some knowledge in the form of certain questions. I hope the session also gives you new insights. So let me just like touch upon the basics again. This is a very, very common structure of nucleotide, and you already know that nucleotides are the building blocks of nucleic acid. So we have a purine or pyrimidine base which is connected with a glycosidic bond with a sugar, and which is a pentose sugar. In case of DNA, it's a deoxyribose, while in case of RNA, it's a ribose sugar. And you can see at the 5th carbon, it is attached with a particular phosphate. So these are the structures of nitrogenous bases. You can see pyrimidine and purines, and these are the major purines of nucleic acids, that is adenine and guanine. And these are the major pyrimidines of nucleic acid, that is cytosine, thymine, and uracil. And these are various bonds which are present in the nucleic acid. You can see in like in a nucleotide. And some of these bonds are very, very important with the structural aspect. So you can see there is a bond which is between the nitrogenous base and the sugar. And can I just also, yeah, the bond which is present between the nitrogenous base and the sugar is actually the glycosidic bond. And then we have got a phosphoester bond which is linking the sugar with the phosphates, all right. Then we have got phosphoanhydride bonds, all right. These are the high energy bonds, which in fact are like, which catalyzes the phosphodiester bond formation and the polymerization of nucleic acid, which are very, very essential in the polymerization of nucleic acid, like nucleotides polymerization to form DNA or RNA. These bonds are also very, very important because they act as like, the energy currency. This is basically the structure of adenosine triphosphate, which we know them as the energy currency. So it is in fact the hydrolysis of these bonds which can provide energy for any particular process to occur in the cell, basically. So these are the deoxyribonucleotides of nucleic acid. I'm going a little bit fast because most of these things are already covered by Kundan sir. All right, and these are the nomenclatures for the same. And these are some of the ribonucleotides, and you can see the structure and the nomenclature for the same, single letter and 3-letter symbols. And you can also see that like the phosphodiester bond is basically linked between the 3' OH and the 5' phosphate in a particular nucleic acid. So you can see the directionality of the synthesis is always 5' to 3', and this particular directionality is actually conserved across the domain of life. Okay, so let us touch upon the first question, that why DNA replaced RNA evolution. So, as Kundan sir already mentioned initially, Professor Thomas Shack, he received the Nobel Prize for the discovery of ribozymes. So, for a very long period of time, people were thinking that, how the life evolved, and it was known from some evidences that DNA could not have initiated the life, because DNA does not have any catalytic activity. When the life wasn't there, we needed some catalytic activity, and RNA was capable of displaying the catalytic activity. Some molecule would have been needed that could carry the genetic information, as well as it could do some kind of enzymatic activity. This is important because if the reactions had to happen on their own, they are going to take a lot of time. All right. So for the reaction to occur a little bit faster, all right, which is very much important to initiate life, like non-randomly, if they had to occur, then some kind of order was supposed to be formed, some kind of constancy was supposed to be formed. And people hypothesized, some great researchers of that time, they hypothesized that RNA could be also having the enzymatic activity, but there was no proof. So in the 1980s, Professor Thomas Shack and his team, they were able to identify RNA having catalytic activity in form of introns, all right, self-splicing introns. So, but the question remains that if RNA is such a beautiful molecule that which is having catalytic activity, along with that which is also having capacity to store the genetic information, then why was it replaced by DNA? So if you know DNA right now, it does not have any catalytic function. It is just there to store the genetic information. So the answer comes from this particular slide, that if you know that RNA has the ribose sugar, and the 2' OH group of ribose sugar, it is very much important in the catalytic activity of RNA. If you know a little bit about self-splicing introns, it is in fact the type 2 intron. That this 2' OH is involved in the nucleophilic attack for which is involved in the splicing reaction. But at the same time, this 2' OH is also very damaging for the nucleic acid itself. This 2' OH can also do nucleophilic attack on its own phosphodiester bond, which can lead to the cleavage of the phosphodiester bond, leading to the degradation of RNA. This makes RNA extremely unstable, which is actually not a good thing if we wish to have a molecule to store genetic information. We want genetic information to remain for a longer period of time, which can be stably sent into the next generation. So in the process of evolution, when I'm talking about evolution, there are two types of evolution, that is chemical evolution and the biological evolution. So specifically talking about the chemical evolution, the evolution that took place before the emergence of life, a lot of reactions were happening. And I was also able to listen Jack Szostak's lecture recently. His lab is working ferociously on the origin of evolution, and he said that around 4 billion years ago, we had such kind of nucleic acid molecules which had a mixture of ribose and deoxyribose nucleotides, ribose and deoxyribose containing nucleotides. So it is pretty much understandable that during the chemical evolution, somehow some kind of reaction basically removed the oxygen from the ribose sugar, making it deoxyribose. And that kind of reaction was actually very much favorable, because the molecule that formed henceforward, it did not degrade. It showed a lot of stability. So it remained for a longer period of time. And slowly and gradually, the molecules which contained more amount of deoxyribose kept on getting accumulated, and they were preferred for acting as a genetic material. So far, we also know that RNA could also maintain its dignity and also continue to stay along with the DNA. for several reasons, which probably it will be very difficult to cover in this lecture. But this is one of the reason that why DNA replaced RNA in the process of evolution. And this particular process aggravates much higher in the alkaline condition. All right, so that's what this particular slide tells you. So I will be happy if someone has any kind of questions in between also. Sir, should I also take up some questions right now, or should I take it at the end of the lecture? Should I take some questions right now, sir?
Yes, sir.
Yeah. So I just wanted to know that if I can take some questions right now, or shall I take it at the end of the lecture?
No, you can. It's all up to you, sir, as you decide.
Definitely. So I'll ask for your questions in between. If at all, we can take more questions at the end of the meeting, but just in case if anyone has some instant questions, they can ask me right now. Or you can probably put it in the chat box. It will be great.
Fine.
Sir, I have a question that why introns still persist in DNA if there is no use?
Okay, so no, it's not like that they don't have any use. Definitely, see, nature will keep something which is useful. It is not going to just go away. If introns exist, definitely they have certain use. But let me not discuss it right now. Let me discuss it at the end of the lecture. Is it okay? Because that will take us away from our present topic. It's a very beautiful question, by the way.
No problem, sir.
Yeah, definitely. So, any questions related to this particular thing? If no, then we can proceed ahead. Okay. Sure. So, this is the first question that we have dealt, and now let us talk about the second question, that why thymine replaced uracil in DNA. So, basically, if you look at uracil and thymine, I don't basically look at as two different molecules. I just see thymine as a molecule which just has an additional methyl group. All right, if you methylate uracil, you are going to just make a thymine. So now, what was the reason? See, there is one reaction that occurs in day-to-day basis, even when you are listening to my lecture, that is a deamination reaction, which is a spontaneous reaction, due to which lot of cytosins will get converted into uracil in your DNA. Now, this is not a rare reaction, as I just said. It happens 100 times per cell per day. This is a spontaneous reaction. Okay. Now, think it of this way. When the evolution was occurring, certainly the DNA repair system was not, formed. And, again, if cytosine gets converted into uracil, and if uracil was already the part of the DNA, if the uracil would have been the part of the DNA, then it would be difficult to anticipate that which was an original uracil and which was the uracil that was deaminated from cytosine. And such kind of changes, obviously uracil can complement with adenine, and cytosine complements with guanine. So that is going to create a lot of mutations, and you will not be able to maintain the genetic information. However, if you don't have in fact, thymine is nothing else but uracil with a methyl group. If you keep a stamp, this is the stamp. If I put a stamp onto the uracil, that is a methyl group, and it becomes thymine, then the deamination of cytosine is not going to make thymine. It is going to make the uracil. So it is going to be very easy for our repair system to understand and identify that here the mutation had taken place. Fine. So this was one of the reasons that, like, the thymine was replaced. Thymine replaced uracil in DNA. Understood. So this is yet another question that now obviously, there are many of the keto forms, like, tautomerization that keep on happening, all right, in the DNA as a part of normal, chemical processes, all right. So yet another question. So this is the question that I wish to focus more in today's lecture, that why nucleic acids are never synthesized in 3' to 5' direction in the biological system. Okay, so we are talking about the biological system. When we are talking about the RNA world, the RNA world is something like all the reactions are occurring in the sea at infinite dilutions. So anything would have been possible. But what is the optimal thing? The things which can, like, fit the thermodynamics, which can take care about the rate of reactions, and which can have certain stability. When I say stability in the sense, like, the one which is preferable for the Earth and that day chemistry. Okay, so can you just give me a moment? Let me just fit it. So there are two answers for that. One is the proofreading activity, but the proofreading activity evolved much later. Okay, just a minute. If you can give me a. Yeah. Can you see the black screen right now, guys? Everyone?
Yes.
Yes, sir.
Yeah. Good. Okay. So if you look at, let me just, like, bifurcate. Let's assume that even the 3' to 5' direction synthesis occurs. All right. So on the left side, I'm going to, give the 5' to 3' synthesis. Let's say that, like, this is a normal nitrogenous base, which is attached with a particular sugar. Okay. Which is CH2. All right, which is attached with a phosphodiester bond above, and it has so let us assume this is a sitting nucleotide. Okay, sitting nucleotide. Why am I calling it a sitting nucleotide? I'm calling it a sitting nucleotide because this nucleotide is already attached to the through the phosphodiester bond to the upper nucleotide, and then you have a flying nucleotide. Okay, the flying nucleotide is carrying its 3 phosphates. Again, please have the knowledge of this, that the 3 phosphates are absolutely essential for the formation of phosphodiester bond in the physiological conditions. Fine. So we are talking right now about the physiological conditions. So this is the RNA nucleic acid that we are going to talk about. So what generally happens that when such kind of things, when the flying nucleotide, this is a flying nucleotide. Okay. So what happens basically when the flying nucleotide approaches the sitting nucleotide, at that time the 3' OH group gets deprotonated with its negative charge, and this does the nucleophilic attack on the innermost phosphate. All right. Which will also be, like, cooperated with the hydrolysis of these phosphoanhydride bonds, the energy of which will be supplemented for the formation of phosphodiester bond, and because of which you can see, because of which you will have the phosphodiester bond being formed between the 2 nucleotides. All right. We will remove the negative charge later on. Okay. Obviously, because these phosphates will have negative charges, and there will be a difficulty for this oxygen to attack the innermost phosphate. So magnesium 2 plus ion acts as a cofactor in taking care of the negative charges which are on the phosphate. So, but I'm not going into detail of that. I'm just showing you the normal reaction, and you can see now even this becomes a sitting nucleotide. Okay, so again you can see that the 3' OH group is now again ready to take on yet another flying nucleotide. Fine, with the same process. So what you see that oops, I think I messed up. Just a minute. Alter. Yeah. I messed up. No problem. Let me just. Please just give me a moment. I'm really sorry I messed up. Okay, I'll quickly draw. What basically you see here is that the directionality will be always in the 5' to 3' direction. Okay. Okay. What do you see? Again, this OH group is now ready to take on another flying nucleotide. You can see the directionality is always in the 5' to 3' direction. You can see at the bottom there will be always OH group. The top there will be the 5' carbon group, the 5' phosphate. So fifth carbon basically. The bottom you will have the 3. So this is a good thing. Now let us imagine. So this is what was 5' to 3' direction, which is known and which is acceptable. Let us look at the reverse. What is the reverse? The reverse is you have a sitting nucleotide. Let's call this a sitting nucleotide. Okay. Sitting. Okay. Let's call this a sitting nucleotide. The sitting nucleotide is having the 3 phosphates. Okay. Having the 3 phosphates and the flying nucleotide okay, which is again carrying the 3 phosphates. The flying nucleotide, let's call this a flying nucleotide in B. I'm calling it a sitting nucleotide because it's linked with the phosphodiester bond below. It's sitting, and this is flying nucleotide. The flying nucleotides, 3' OH group needs to attack the innermost phosphate after getting deprotonated. Okay. So here, look at this particular case. Here, the sitting nucleotides OH group was attacking the flying nucleotides innermost phosphate. On the contrary, when you look at the 3' to 5' strand synthesis, at that time the sitting nucleotide is showing its 3 phosphate, and it's the flying nucleotides 3' OH group which is going to do the nucleophilic attack onto the innermost phosphate. So that is good, not a problem. Everything is fine. All right, it will do that. Let me just rub it. Okay. So this time, the hydrolysis of the sitting nucleotides 3 phosphates are going to take place. Instead, the flying nucleotides 3 phosphate, you will have hydrolysis of these 3, and then you can definitely form a phosphodiester bond. Okay. Please excuse me for my drawing. Nitrogenous base. Okay, you have to hydrolyze these bonds, not an issue. Again, the magnesium 2 plus ion is going to act as the cofactor in the reaction. So you can see what is happening. Now look at the direction of the synthesis. The direction of synthesis is in this way. Okay, from 3' to 5'. Again, now this will be again now new sitting nucleotide. Okay, this is the new sitting nucleotide. So this new sitting nucleotide is again ready to take on the new flying nucleotide through its 3' OH group attacking the innermost phosphate. So chemically there is no problem. All right, whether it happens 5' to 3' or whether it happens 3' to 5', doesn't there is no issues. In fact, there is no issue. All right, in the mechanism of reaction, but there is an issue with thermodynamics. Okay, now why am I calling it an issue with the thermodynamics? When I'm saying that these phosphoanhydride bonds, these phosphoanhydride bonds are very labile. Like they can get hydrolyzed very easily, and that's a spontaneous reaction. All right, the hydrolysis of a phosphoanhydride bond is going the delta G, the standard delta G value is minus 7.3 kilocalories per mole, or 30.05 minus 30.05 kilojoules per mole. So it's a spontaneous reaction. Water can easily hydrolyze this phosphoanhydride bond. So you have to remember, even if one of the phosphate is hydrolyzed, then this reaction it's impossible for this reaction to occur. Okay. Now imagine the hydrolysis of this phosphoanhydride bonds is a very natural thing. It keeps on happening. Even in your body, some of not all the ATPs would be probably completely utilized. It might happen so sometimes that some of the phosphates gets hydrolyzed, and that ATP again needs to be recycled to make ATP. All right, because everything is happening in water-based cell, and so that's quite possible, right? So if when the 5' to 3' synthesis is occurring, and by chance if the new nucleotide comes, all right, the flying nucleotide comes, and it might happen so that one of the phosphate is hydrolyzed. Okay, so this reaction will not occur. The phosphodiester bond formation will not occur. Okay, but it doesn't matter. This particular OH group, 3' OH group, will wait for a new nucleotide to come and join. All right, it can wait. So this will go away for the recycling or whatever it is. It will wait for say for example, half the way suddenly, because see, the sitting nucleotide is having the responsibility of showing its 3-phosphate, and half the way one of the phosphate got hydrolyzed. All right. So will that sitting nucleotide be able to accept the new nucleotide? It will not be able to accept the new nucleotide, which will lead to the truncation of the entire synthesis. The entire strand will get wasted. It will stop here. The synthesis will stop here, which will again create a huge waste and huge energy waste. Basically, when we are talking about cell, we have to talk in terms of energy utilization. So that's going to be a huge waste of energy, because you can see half of the way everything was fine, and then the strand got truncated because this one of the phosphate got select this particular vulnerable process. Thermodynamically, this was not a favorable process. Okay, and thermodynamically, 5' to 3' was a favorable process. So this is something that got selected. This happened. See why this thing is very important, because our entire living life is actually sitting over the formation of phosphodiester bond. It is actually sitting over 5' to 3' direction strand synthesis. The entire biochemistry or everything is evolved around the central dogma process. So this is that's why this particular reaction is having that great importance. All right, guys. Okay. So the next reason is obviously, now let us come into the DNA protein world in across prokaryotes and eukaryotes. We are now talking about the something which is related to proofreading activity of DNA polymerase. Is it why even 5' to 3' direction synthesis is preferred for the proofreading according to the proofreading. So I hope you all would have had a little bit of the knowledge regarding the replication process and the proofreading activity of DNA polymerases. If not, it will be covered in the later lectures by some of the experts. But still, see the proofreading as the mechanism of DNA polymerase in which it can actually correct the wrongly incorporated nucleotides. Fine. So generally, what happens is think it in this direction. First of all, look at the right side. All right, the process B, which is the actual 5' to 3' direction synthesis. So what happens? You can see at 5' you have got a phosphate, and 3' you have got an OH. So this is the flying nucleotide. Flying nucleotide will come with its 3 phosphates. 2 of the phosphates will be removed. The energy will be utilized to form the phosphodiester bond. So you can see this particular nucleotide has joined with this, and it has also become a sitting nucleotide. Now, say for example, if this was a wrongly incorporated nucleotide, obviously the complementary strand is not shown here. Only single strand is shown here. But say for example, here it was adenine by mistake. Instead of thymine, say for example cytosine came by mistake. Okay, not going into much detail right now, but think it in that way. So once the phosphodiester bond has been formed, so it's a commitment stage. Now it has to be removed through some chemical reactions. Right? So the chemical reaction that I'm talking about is the Exo Nucleus Activity, which is 3' to 5' Exo Nucleus Activity. If the synthesis is occurring in 5' to 3' direction, and if you wish to cut the DNA in the reverse direction, so that is 3' to 5' Exo Nucleus Activity, which is also there with the DNA polymerase. So it will remove the wrongly incorporated nucleotide. Again, this 3' end will be free with its own OH group. All right. So again, the correct nucleotide can come and join, and it can do the synthesis. See, the burden of energy is with the flying nucleotide. Okay, that's why this particular process is very favorable. But if you look at the hypothetical 3' to 5' strand growth, all right, why this particular process is not favorable, let us see. So in the 3' to 5', the burden of energy is with the sitting nucleotide. You can see this is the sitting nucleotide, the 3 phosphates, and the flying nucleotide will come with its OH group. All right, so this OH group will do the nucleophilic attack on the innermost phosphate of the sitting nucleotide, thereby releasing these 2 phosphates. Got it? And you can see very nicely the phosphodiester bond has been formed. But what if this particular nucleotide is wrongly incorporated nucleotide? So obviously it has to go under proofreading. So if the directionality of synthesis is 3' to 5', in this case 3' to 5', then the proofreading will be in the opposite direction, that is 5' to 3'. So you are going to remove this particular wrongly incorporated nucleotide, and you can see it is being removed. But now look at it. What is remaining here? Only 1 phosphate is remaining here. Why only 1 phosphate? Because in the initial step only it had given off its 2 phosphate for the formation of phosphodiester bond. With 1 phosphate, the new incoming nucleotide cannot form phosphodiester bond because the burden of energy is with the sitting nucleotide, and that's why this strand will be truncated. If once the proofreading will occur, this strand will be truncated, and there will be again a lot of waste of energy and so as and so. Okay, so that's why according to the proofreading also, this particular step is not preferable. So this was something which was related, that why it is always that it's a universal truth that the nucleic acid synthesis occurs only in 5' to 3' direction. Okay. So yeah, this is the same thing. This is the explanation of thermodynamics, but I have already explained it in my diagram. Okay. So yet another very interesting question, that why nucleic acids contain only and only pentose sugars and not other sugars. So I was actually supposed to take your lecture today on structure of aldoses and ketoses. And I have developed some technique to derive the structures. So you can actually make 84 structures of sugar, that is Fisher and Hoare projection. But that takes a lot of time in some practice. So we turned into this particular presentation, which is also a part of biomolecules. So this is a very important question, that out of all the sugar, like you have glucose, you have got xylose, lysose, allose, aldose, all right, so many things are there. You have got trioses, you have got tetroses, you have got pentoses. Within pentoses, you have got ribose, arabinose, xylose, and lysose. All right, and then you have got hexoses. All right, allose, aldose, glucose, mannose, bullose, iodose, soase, and so on. So many of them. Galactose, tallose. Also, the D and L form, dextrorotatory and leo-rotatory forms. Okay, out of all these sugars, why the nucleic acid specifically, selected ribose to be its part? So this is a very important question, fine, because nature was forming a very, very important molecule to store the genetic information. Fine? So that's why this question is very important for that now, with that perspective. Okay. So you can see again, I'm showing you the same slide, that you have got all these nucleotides are having riboses, deoxyriboses for DNA and riboses for RNA. Now let us take this question one by one. So I have taken this information from a paper which was published in year 2003, and if required, I will also share the link with Amit sir. So probably can dissipate this particular paper to you all. So I've taken the important points which is actually discussing that why ribose was selected. Okay. So first thing, that why not triose or tetrose in nucleic acid? Okay. To look at a particular triose sugar, the aldotriose, that is glyceraldehyde. So right now we are talking about D-glyceraldehyde. So let us say that the D-glyceraldehyde would have been selected as the sugar instead of a particular ribose. You can see that the place for affixing the phosphate would be at the third carbon, and the place for affixing the nitrogenous base would be at the second carbon, and that would have created a lot of steric hindrance, which is not preferable. And statistically also, experimentally also, it has been demonstrated by some of the intermediate molecules that such kind of combination is not preferable for the polymerization process. It is very, very difficult to polymerize such kind of molecules which is having triose sugar, so triose nucleotide. Okay, another thing is that why not tetrose, by the way? Okay, so what happens that this OH group, the placement of this OH group is in a very, very close proximity to this particular phosphate. Okay, so if we try to polymerize it with the tetrose, obviously the process of polymerization is extremely slow compared to when we have ribose sugar. But still, if we are placing the triose sugar as in the structure of nucleic acid, this particular OH, because of its close placement to this phosphate, is going to show a very, very high nucleophilic attack possibility. Okay, and that is going to degrade the polymer. As the polymer is getting formed, the degradation also starts occurring. Fine, so stability is in a very, very big question. Okay. So another question is that why not hexose sugar in nucleic acid? Okay. So obviously, it is not that hexose sugar cannot be a part of the nucleic acid, but there will not be any kind of constancy. Fine, because you have got multiple OH groups. So these multiple OH groups are placed, since right now we are looking everything in the two-dimensional, but if you try to visualize these sugars in 3 dimensions, then you can actually understand the bond angles, bond lengths of all these OH groups, which are flanking around. Lot of OH groups are there. So when the polymerization, there are various possibilities that happen. The phosphodiester bond formation can happen at any of these OH groups. That is one thing. Another thing is that because of the presence of these multiple OH groups, it can create a lot of problems with the random nucleophilic attacks. which in the long run is going to create a very, very unstable and unfamiliar nucleic acid molecule. You just imagine the nucleic acid molecule in which some of the phosphodiester bond are there at the 2' carbon, some of them are at the 5' carbon, some of them are at the 3' carbon. All right. So that is going to create a very, very vulnerable, very, very distorted structure of nucleic acid. Let's say for RNA only, because all these selections and all these things happened in the process of chemical evolution. Okay, so that's why also it was not. So whatever I'm trying to tell you, I'm just like giving you the gist of the experiments, and then even though within the pentoses, why only ribose, and that too only why only beta ribose. All right. So obviously there are two. If you know that there are two anomers, alpha and beta anomers are there. Both are anomeric forms. So in that also, why only beta D ribose? Why not alpha D ribose? And also why not other pentoses? Okay. So please try to understand this very, very specifically. Okay. So you have got alpha D ribose, you have got beta D ribose. Now look at the placement of the OH groups. All right, it's facing down, it's facing up. Obviously, just try to visualize it in a three-dimensional space. So what happens is basically when you have got the OH group which is facing above on the top. So obviously the glycosidic bond will form here with the nitrogenous base NB. So nitrogenous base is a very, very bulky group, all right, so it can comfortably, so you can see it is far from all other, attacking groups. So at a very, very comfortable position. If you look about the alpha position, so the glycosidic bond would be formed here, all right, and which is going to create a lot of problems. Problems like steric hindrances, or probably the problems like, the hydrolysis of this nitrogenous base. Ultimately, the rate of polymerization is extremely slow, or it is defaulted in such kind of cases. So alpha D ribose is not preferred in that way, and for the same matter, none of the alpha D sugars are preferred for that matter. Okay? None of the alpha anomers are preferred for that matter. For the same reason, we have another thing, that is alpha D Arabinose or beta D Arabinose. So you can see what's happening in case of alpha D Arabinose. Say for example, if your nitrogenous base is affixed here, all right, nitrogenous base is affixed here, you have got an OH group which is again in the close proximity of this nitrogenous base. It kind of creates the problem. Okay. Another thing, again here also the same way, you have got an OH group, you have got a nitrogenous base here, which is also again in the very, very close proximity of creating problem. Okay. So what I am trying to tell you is we actually have to compare the thermodynamics with the standard molecules, and the standard molecule is performing the best out of all these combinations because of the steric hindrances or because of the nucleophilic attacks. And then we have got alpha D xylose, then we have got like beta D xylose. So obviously you have got an OH group which is very close to the phosphates near the 5' carbon. All right, again OH group is very near to the phosphates which are near to the 5' carbon. It interferes a lot. Okay, and then also again we have got alpha D xylose. Okay, we have got alpha D xylose. So for certain matters, I already discussed that alpha D anomers will never be preferred. But you can see again the same issue. You have got this, you have got this. This is a problematic molecule. All right, and you have a lot of this and this as a problematic molecules. Okay, so that's how. See, to summarize this particular thing, see the rate of polymerization was found to be the best with the beta D ribose compared to all other of these sugars. Some of these sugars did polymerize, however, the polymerization was slow. Some of the sugars did polymerize, but they were unstable. Like the nucleic acid was pretty much unstable. That's why beta D ribose was selected in the process of chemical evolution. These are hypotheses, by the way. No one has actually gone and seen there that what actually made these sugars to get incorporated into nucleic acid. But these are very strong chemical and substantial evidences that why other sugars were not selected. And obviously, as Naresh sir said, that there are certain mysteries that we can see only and only L amino acids and D sugars in the living system. This is an enantiomeric bias. All right, that there is a chiral bias in the living system. People are just trying to hypothesize why it is like that, but you can see in most of the biomolecules, majority of the biomolecules and majority of the dominant sugars, we have D sugars, and in the proteins are made up of L amino acids. All right, so again, but let me also tell you that if you can chemically make the nucleic acid containing the L sugars, it does polymerize with a comparable efficiency of the D sugars. All right, it does happen, but the system was made to be formed with the D sugars, so that's out of the question. All right, so now I'll be taking my last question, that is why DNA double helix is anti-parallel in nature. So this is how you have to look at that, how basically the nucleic acid is stacked one, like the various base pairs are stacked with each other. You can see this z-axis, you can have a base, you are having base pair. In the xy axis, you are having the sugar. So they are arranged in that way, and you can see they are arranged in a spiral form. All right. So the anti-parallel nature, and you can see between these two bases in a base pair, whatever you can see, these are the hydrogen bonds, right? See, the hydrogen bonds are very effective only at a certain particular distance. All right, if you increase the distance, the effectiveness of the hydrogen bond becomes very less. So in the normal Watson and Crick base pairing, all right, so this is basically a normal Watson and Crick base pairing. You can see that the hydrogen bonds between adenine and thymine, there are two hydrogen bonds, and you can see the length of them, 2.8 angstrom and 3 angstrom. All right, and in GNC you can see 2.93 and 2.9. So all these are pretty much equal to 3 or less than 3, in which like the hydrogen bond can actually execute an effective interaction. All right, this happens only when the bonds are, the DNA is in the anti-parallel state, 5' to 3', 3' to 5'. All right. Look at if you try to basically make parallel DNA, with probably you can make it in the lab at certain places in the genome. You also see hexene base pair. What you see is that most of the hydrogen bond interactions, because of the stacking and because of the sterical hindrance and because of the base stacking, as I just mentioned, these hydrogen bonds are going much above 3.5 angstrom. Okay, 3.5 angstrom, which is not very much effective distance for the hydrogen bond interaction. So thermodynamically only, when you put two strands of DNA inside the solution, they are going to arrange themselves so that the best kind of interactions can occur, and they will zip themselves in the anti-parallel fashion, because that is going to give them the best hydrogen bond interaction. Okay, so that is going to create some improper stacking. You can see how improper stackings are there. Some of the visualization of how the stacking would occur if you have got parallel strand synthesis. But thermodynamically, it is less favorable. Okay, so that's all from my side, sir.
Yeah, Trivedi sir.
Yes, sir.
Yeah, fine. Actually, we don't have light since morning. Getting it, and
okay.
Around 14% battery left. So I thought I should give you reminder, but you concluded on time. That this is very good, sir. But I can give some time to student if they have any question, 1 to 2 question, then they can ask. Dear student, if you have queries, you can ask to sir. Monica, sir, please, sir,
okay.
They have sent some queries. Please see that.
Okay, so sir, please let us know how these topic questions are asked in competitive examination with example. Okay, So, the topics like, what kind of anomer is present in the nucleic acid, like DNA or in DNA and in RNA. Like, as I simply mentioned that you have got beta D form of deoxyribose, or you have got beta ribose in, the RNA. All right. So such kind of questions. And also, like, I probably will submit some of the MCQ questions to Amit sir. So that will be also a good practice. So this is just one example. Okay. So Mahim Dave is saying, what is the reason for Google-based pairing? Oh, okay. So this is so you are taking me towards translation. So you have asked me two questions, Mahim. That why if the introns are introns are actually going to get removed, then why are they there? That is one question. And another question is that what is the reason of Google based pairing? So please try to understand this. The Google based pairing is different from Watson and Crick based pairing. When the tRNA and mRNA, they actually fit inside the ribosome, and when that codon anticodon match occurs, at that particular time the structure is such that, like, the third, that is Google base pair. See, the name itself suggests that googling, basically that interaction between them is not so strong. All right, and that has actually given us the competitive edge, that like we have 20 amino acids only, but we have got 61 amino acid coding codons. All right, so you can have few of the amino acids can take more than one code to code itself, right?
Fine. So I can allow one more question only, because your nearby battery is about to exhausted.
Right, sir.
And if you have any query question, you can ask directly to sir on his mail ID.
Yes. Actually, I will give a very good explanation for because the time is less. I need some time to explain his question regarding the introns. But definitely, Mahim, you can definitely be in contact with me. I will explain. It's a very beautiful question. It's my favorite, actually, topic.
Oh, that's great.
Yes,
sir, you explain everything in so nice way that each and every student has got each and every concept very clear. Whatever question you raise and answer you given in nice way. I also learned some new things from your lecture, and really it's so far competitive question is concerned. Really, sir, this particular lecture is fruitful and useful for a student, not only for GAT-B, but this will be also useful for CSIR NET examination. And behalf of GSBTM and NV Science College, sir, we thank you for sparing your valuable time, and in your future, we will also contact you for more lectures so that a student can get more benefit. So thank you very much, sir, for your nice lecture, informative and excellent lecture so far competitive examination is concerned. And now I am going to bind up this program, and rest of the information will be communicated by Amit sir. So Amit sir, would you like to say something?
Yes, sir. There is a query by Mr. Shashikant Shukla about the PPTs. So already in the morning I have told that by 4 p.m. all the PPTs will be there, and whatever the links of the lecture, they are already there on YouTube. So you can also recap those things. Ujwal sir, have you this particular PPT, can you share with us, sir? So
definitely, sir. Definitely. So, because in the morning only I took this decision that I'll change my topic. So otherwise I was actually going to take what we say, a practice kind of session in which there was no need of PPT, but this particular PPT I will share, definitely.
Yes, sir. So dear student, by 4 p.m. today you will receive all the PPTs which we have received from the resource person, and we will also tell you the time for a practice test. So be ready with these PPTs, and then the questions when we receive from the resource person. So whatever topics you have studied through these GujBT lecture series, from that only we will conduct a mock practice test. So as few students were asking about what type of questions are being asked, So that will be a live for you, and those each question will be a fact in itself. So be ready for the test also. And I request you to take all these sessions with utmost importance. And if there is some reason for you not to attend, then only you miss the sessions. Otherwise, you have are very disciplined students. Thanks a lot for being there. And Ujwal sir, you also, I thank you a lot. And I think there are 2-3 sessions still. We want you to be there, particularly the sugar things and other which are left. So
Definitely, sir.
you know, sir.
I'll be really happy to teach the structure of sugars, nucleic acids, amino acids, everything, sir.
It will
Thank
be
you.
nice, sir, because questions are there. On
Definitely,
these
sir.
particular
Definitely.
things.
Okay.
Definitely, sir.
Okay,
So
then.
thank you once again, sir. Sir, now I'm going to left it. Okay, sir.
No, no,
Sir?
sir.
Okay, okay.
Okay, sir. Okay, sir.
Thank you.
Okay. Thank you, everyone. Thank you, students, for your patience.
Thank you,
Amit,
everyone. Yes, sir.
sir, Amit sir,
Yes,
बंद
sir. Now
करता
we are
हूँ मैं.
concluding only, sir. With we are
Okay.
ending the meet, sir.
Okay, thank you, sir.
Okay,
Thank
Kulan, sir. Thank you, sir.